Good evening, everyone. We have hundreds of people that are probably already on the webinar and that are supposed to join us from all over the country, from Washington to Texas to Miami. A big thank you to everyone that's here, especially those of you who have organized something of a watch party for your local group. Tonight's webinar comes to you by the Christian Medical and Dental Associations, CMDA, as well as Erskine Seminary and the Discovery Institute. My name is Grant Hewitt, and I am the Southeast Regional Director for CMDA. More importantly than that, you'll soon learn that our speaker tonight is a very interesting scientist and Christ follower, and that's Dr. James Tour from Rice University. I could hardly be more optimistic about our time, in that I consider Dr. Tour to be an authority on the relationship between faith and science. And after this webinar is over, I'm confident that you will too. Most of our attendees tonight, not all, most, though, are currently studying in a field of science such as dentistry or medicine. So you'll have plenty to consider and discuss afterwards. Our topic tonight is important because it is far too common today to understand faith, especially a committed faith in the Bible, to somehow be at odds or in conflict with science. Faith and science just don't seem to mix for some reason, at least in many people's minds. At least since Copernicus and Galileo, Christians have wrestled with these matters. More recently, the late Stephen Hawking boldly stated that one can't prove that God doesn't exist, but science makes God unnecessary. Another example would be the Nobel Prize-winning physicist Steven Weinberg, who asserted that one of the purposes of science is to free people from religious superstition. So that's one of the purposes of science, apparently. Frankly, we need people in the academy who can and will counter these kinds of opinions head on. And thankfully, Dr. Tour is just such a person. I am so grateful that he has chosen not to be silent on issues of faith and science and not allowed related false claims to go unanswered. Dr. Tour was quoted in Lee Strobel's highly influential book, The Case for Faith, as saying, quote, "I build molecules for a living. I cannot begin to tell you how difficult that job is. I stand in awe of God because of what he has done through his creation. Only a rookie who knows nothing about science would say that science takes away from faith. If you really study science, it will bring you closer to God." Our hope tonight is that this presentation will bring you closer to God. For those of you who may not be familiar with him, it really is important that you know where his comments and where his talk tonight is going to be coming from. And so I want to give you a small sliver of his resume. And so here's an excerpt from his CV. Dr. James Tour has a PhD in organic chemistry, a postdoc at Stanford University, and voted one of the top 50 most influential minds in the world. He was a visiting scholar at Harvard University, has over 650 research publications, and voted the R&D magazine Scientist of the Year. Dr. Tour is in the National Academy of Inventors. He's a member of the American Association for the Advancement of Science. He has over 120 patents and started 8 companies. He works in areas ranging from medicine to medical devices to computer memory. He also works with these fascinating machines that he calls nanocars, which I hope he will at least mention tonight. Any introduction of Dr. Tour, honestly, would be incomplete without stating very clearly that he is a devoted follower of Jesus Christ. This is a man who is not afraid of controversy and is not embarrassed to be a follower of Jesus. Thank you so much for being here, brother. The floor is yours.
Thank you. Thank you for inviting me. And I'm going to open up and start sharing some slides here. All right, so I'm going to talk about nanotechnology in the beginning and then a critique of the current origin of life research. This kind of is a composite of some of our research. We've developed something called laser-induced graphene. This is where we can convert any carbon material into graphene with a quick laser pass. This is the same type of laser that's used in any machine shop. You can convert the carbohydrate strands on bread into graphene. This is not dropping graphene on top of it. This is a conversion of the carbohydrate into graphene. This is probably going to lead to 5 different companies. We've learned how to split carbon nanotubes to make graphene nanoribbons. I'll show you an application of this in a moment. This is already 1 company, but it's going to go into 5. This is already sold by Sigma Aldrich, which is now Millipore Sigma. This is computer memory. This is now a public company. We're developing a drug for traumatic brain injury and stroke and dementia. That's a company that will start up very soon, where we could take a brain that would normally look like this after a traumatic brain injury and have it look more like that. This is where we have carbon nanotubes fused to graphene. This has led to a battery company called Zeta Energy. This is graphene quantum dots. This is in a public company now. Lots of applications of this. And in fact, one of the more recent ones that they come out with that has pure approval in Europe, in the EU, and soon to have approval in the US, I hope, is where we can do rapid COVID tests, 200 tests in 15 minutes. So you could do even a whole plane load of people waiting to get on an airplane. And it's, excuse me, it's all colorimetric based, and it's more accurate actually than PCR. And that's been shown. So, but we have to always compare against PCR. This is where we've taken plastic waste and we use it to trap carbon dioxide. So 2 big problems in the world, plastic waste and carbon dioxide. And we use one to address the other. We have a new process here, and that's to take any carbon feedstock and to convert it into graphene in a bright flash. And that's a new company that's just starting out, which is the fastest growing company I've ever been part of. It's called Universal Matter, and really taking off. And so we put a high voltage across this, and the high voltage, high current, and it breaks every carbon-carbon bond, and it reforms as graphene, the most thermodynamically stable form of carbon. This is our nanocars, which Grant asked me to mention, and it's here. We built these little nanocars, and they have motors that you can shine a light, and they spin 3 million rotations per second, and will drive these cars forward. We won the first international nanocar race, which was in 2017, by a lot. And then the next race is scheduled to be in 2022. This is where we've taken the motors of these nanocars. We put on peptide add-ends, and we could target it to specific cell surfaces. These then drill in through the lipid bilayers to form holes in cells. And we have several cancer efforts going. This is a superbacterium. We're killing several different types of superbacteria. It just destroys the cell wall and goes in and just gelatinizes the innards. And so they're very good at killing. This is 3D printing graphene objects. This is a rat that has had its spinal cord completely cut in 2 at C5 of the base of the neck, complete transection of the spinal cord. Then we put 1 drop of graphene nanoribbons, a 1% solution of graphene nanoribbons in polyethylene glycol. And after 2 weeks, that rat gets up and starts walking again. And so here it is after 2 weeks, getting up and walking again. Scored a 18 out of 21. 21 is the optimal mobility after 2 weeks. And then after 3 weeks, scored a 19 out of 21 on a mobility scale. So what happens is the graphene nanoribbons provide conduits, billions of roadways for the the neurons to reconnect between the top and the bottom. This is after 3 weeks, so she's doing rather well. And this is just spun off into a company to try to develop this further for the reconnection of spinal cords and optic nerves and peripheral nerves. This is a listing of the different companies that we've started over the last 6 years, DOTS and Weebit being public companies now, the battery companies, the neurocords, the graphene nanoribbons, Universal Matter, which I mentioned, the nanorobotics is the molecular nanomachines to drill into cells, Exarient. This is going into phase 2 this fall for treatment of pancreas cancer. LIGC, this is this laser-induced graphene to make across many different surfaces. H2 Blue, this is for CO2 capture, texting tip, Roswell, and a few others. And then these are a bunch of other companies that we hope to start in the next 6 to 8 months. Okay, with that, let's start talking about abiogenesis. So abiogenesis is the origin of life from non-living matter. To construct any convincing theory of abiogenesis, we must take into account the condition of the Earth about 4 billion years ago. So these are quotes. So what I write here is, for synthesis to be categorized as being prebiotically relevant, it must use chemical reagents and conditions that are presumed to be available upon an early Earth, or else it's not prebiotically relevant. All right, abiogenesis takes place before biology and before biological evolution. Hence, it is prebiotic or pre-biology. What are the characteristics of life? Definitions are, again, I'm quoting, responsiveness to the environment, growth and change, ability to reproduce, have a metabolism and breathe, maintain homeostasis, being made of cells. Remember that, it's really important, being made of cells, passing on traits to offspring. Homeostasis is the steady internal physical and chemical condition. Origin of life, molecules don't care about life. They just don't care about life. Organisms care about life. Chemistry, on the contrary, is utterly indifferent to life. Without a biologically derived entity acting upon them, like a human being, molecules have never been shown to evolve toward life. Never. This whole idea of chemical evolution, this term, I had never even heard it used in chemistry until the last few years, because molecules don't evolve by like people envision biology evolving. Molecules don't care about life. Almost every chemical synthesis experiment in origin of life research can be summed up by protocol analogous to this. You purchase some chemicals, generally in high purity from a chemical company, mix those together in water at high concentrations or in specific order under some set of carefully devised conditions in a modern laboratory. You obtain a mixture of compounds that have a resemblance to one or more of the basic classes of chemicals needed for life: carbohydrates, nucleic acids, amino acids, or lipids. Almost never are those made in homochiral form, meaning one particular enantiomer. Then you publish a paper making bold assertions about origin of life from these functionless crude mixtures of stereoscrambled intermediates, much like Miller did in 1952. Engage with the ever-gullible press to dial up the knob of unjustified extrapolation. Watch the mesmerized layperson exclaim, "You see, scientists understand how life formed," and encourage a generation of science textbook writers to make colorful, deceptive cartoons of raw chemicals assembling into cells, which then emerge as slithering creatures from a prehistoric pond. All right, here's the synthesis problem. Molecules that compose living systems almost always show homochirality. When building molecular systems, constant redesigns are needed to take the syntheses back to step one. It is often impossible to remove a moiety once it's been added to a molecule. The synthetic reactions do not know how to stop their current course of progression or why to stop. There's no targeted goal. Molecules don't know they're going toward life. They're mindless. Time, although claimed to be the great savior of abiogenesis, can actually be the enemy. For example, carbohydrates are kinetic products, meaning they undergo caramelization or the Cannizzaro reaction for decomposition. A prebiotic system does not have the ability to easily purify the structures. You can't purify, you can't do chemical synthesis because the impurities gum up the works. Reagent order addition is essential. You can't just willy-nilly mix things. They have to be mixed in a proper order, proper time. The parameters of temperature, pressure, solvent, light or no light, pH, atmospheric gases or no gases have to be carefully controlled in order to build complex molecular structures. The characterization at each step is essential for the chemist, but hard in a prebiotic system because it does not know, it knows nothing about molecular structure. How do you go on to the next step when you haven't characterized what you have? Somehow, nature has to do this if you're going to think of a prebiotic world. How does biology do this? Biology has recognition systems that are more complex than the system you're trying to build to characterize these. If it's not the right structure, it is rapidly decomposed because these things would really mess up the systems if their concentrations were high at all. The mass transfer problem will be the killer of all roots. If you've done a multi-step synthesis, you might start with 100 grams of something, and after 5 steps, you have 10 milligrams. What happened? Well, you lost it because of you got 10% yield here, 5% yield here, and then you're down. That's the mass transfer problem. So what you do is you keep a laboratory notebook that tells you how you optimize this, so now you can go back and make more. You have to keep going back to the beginning to make more, to scale it up, to get you further steps down the line. But nature never keeps a laboratory notebook, so it can't go back for more material. These are fundamental problems you got to deal with in chemical synthesis. All right, so if you look at the different classes of compounds, carbohydrates, we'll start with those, which are sugars, sometimes called saccharides, and the polymers therefrom. So here's some naturally occurring monosaccharides. So it's not just one, it's not just glucose. Here's just some of naturally occurring saccharides, and they're all chiral here. And they have more than one stereogenic center. And then you have, here's the homopolysaccharides when they hook up. So cellulose is made up of only glucose, but it has a beta linkage between 1 and 4. Amylose is a starch. It's made up of glucose, but it has an alpha 1,4. I'm sorry, so cellulose is beta, amylose is alpha. Chitin, this is the sugar component, N-acetylglucosamine, and it has the beta 1,4 linkage. All of these are just the homopolysaccharides. You also have mixtures of polysaccharides. They can have different sorts of arrangements. Have 1,6, 1,4, and 1,6. You can have alpha or beta. Hard problems. If you want to make a nucleotide, you have to first be able to make ribose, the sugar that it's on. Well, ribose has 8 possible isomers because you have 1, 2, 3. That's 2 to the 3rd possible stereoisomers. So you have 8 possible isomers. And so you have to control those 3 stereocenters, or else you're going to get all isomers. If you get all of those isomers, you can't do chemistry off of those. The DNA just needs the ribose enantiomer. The RNA needs the ribose. DNA you have to take 1 hydroxyl off of that. Hard to be able to do, to control that. And if you're making saccharides or sugars, if making them isn't hard enough, hooking them together is also hard, because if you just consider D-glucose, D-glucose can be hooked together like this. So this is the alpha. This is this alpha linkage, and this is this connection between this particular hydroxyl group and this particular hydroxyl group. This is your anomeric center here. And so, or it can be hooked together like this, or like this, or like this, or like this. So these are the 5 possible reducing saccharides that you can get, because glucose can assume an open form with 4 stereogenic centers, or a closed form. It can be closed alpha, or be closed beta, or it can be closed in a 5-membered ring form, again, alpha or beta. So you have each has 5 stereogenic centers. So there's 32 possible isomers here that you can deal with. So this is a big, big problem. So even if you have glucose, how's it going to fold and close? And so polysaccharides, if you look at polysaccharides, here's cellulose. It's just glucose, but it has a 1,4 linkage, means the 1 carbon here is hooked to the 4 carbon of this one. That's cellulose. People don't generally eat cellulose for nutrition. You might eat it for fiber, but you don't see people eating grass for nutrition. Because when we eat starch, we're looking for the 1,4 linkage, but the 1,4 alpha linkage, not the beta. We have enzymes that can digest this, not the beta linkage. And so you have to be able to control that. How is that controlled? If you just throw these things together, nobody knows. Now, if you just take 6 saccharides, let's just take 6 nucleic acids. Say you had DNA and you had six of the A bases on your deoxyribose. What are the ways that you can hook those 6 A bases? One way, A. That's it. But if you had 6 glucose molecules, all the same molecules, how many different ways could you hook this together? In other words, this alcohol could hook at this site, say alpha, or this alcohol could hook at this site, beta, or this alcohol could hook, or this alcohol. So there's all these different tentacles on it. Well, it turns out D-glucose, just 6 units of D-glucose, just 6, can hook up over 1 trillion ways, 1 trillion different ways. If you don't get it right, the cell doesn't live. How does that happen without having enzymes to put this thing together? Nobody knows. Nobody knows. Really, really hard chemical problems. So summary, carbohydrate synthesis is exceedingly hard. Coupling of carbohydrates is painstaking. The untrained have no idea how hard the syntheses are to perform. The trained remain clueless. The untrained see little difficulty in conducting these syntheses because they don't know about this. How do you do it synthetically? You have to very selectively protect, deprotect, protect, deprotect, so that if you just want to make a few hook together, it's many, many steps because there's very careful selective protection, deprotection, which enzymes don't do. They can selectively pick these things out. It's utterly amazing. Then if you look at peptides, which are made up of amino acids, and this is what makes up proteins and many enzymes. So you have to have the amino acids. There's 20 naturally occurring amino acids. Almost all of them, not all, but almost all of them have a stereogenic center. This one doesn't, but all the others do. And you have this problem of you have to control the stereogenicity about this center. So unless R equals H, this is going to be a stereogenic center, what used to be called a chiral center. And you have to be able to control that. But not only that, there are active side chains, meaning that there are groups on these side chains that could also do the attack if you didn't first protect them. These are all active side chains. This one, this one, this one. All of these are active side chains that would have to be protected. How you get amino acids to hook together in an aqueous solution or non-aqueous solution without doing side chain protection, nobody knows. So whenever they address this, they will show you they'll show you glycine, or they'll show you alanine, something without the active side chains, because they don't know how to answer this question. How do you do the protection, deprotection? They always leave that out because nobody knows. That's the problem with this. So that when you do solid phase peptide synthesis, you always have to have these side chain protecting group, SPG, side chain protection group. Then you have to protect that nitrogen protecting group so that you can load this on, so that you have to keep the nitrogen protecting group so that the carboxylic acid that's on this end would hook to the nitrogen that's, you've now deprotected this on this end. So there's this constant protection, deprotection going on just to make the polypeptide. And then at the end, you have to deprotect all these side chain protecting groups. This is what happens in a machine. That's why it's been published in Nature that it is inconceivable that sophisticated and coordinated macromolecules suddenly emerge at the origin of life. This guy, Matthew Powner, he is an origin of life researcher. He himself is confessing this. And he says, in practice, there are good reasons why these reactions are ineffective in water. That's the polymerization of amino acids into peptides. They're ineffective in water. They don't polymerize in water. And even if you found an environment in which they polymerize, if you don't have selective protection, deprotection, you're lost. You're utterly lost. It is only those who do not understand chemical synthesis that think that this can happen. Those who understand chemical synthesis are like, I have no idea how this could have happened at the origin of life. Like this guy is confessing. So you look at nucleotides, DNA and RNA. So this is what you have in RNA. DNA is one carbon simpler in the sense that you lose one stereogenic center. So that's DNA. DNA is going to be a lot more stable than is RNA. And so how do you do solid phase DNA synthesis? It's constant protection, deprotection. The problem is you have an active hydroxyl group here, you have an active hydroxyl group here, but you got to make this into a leaving group, and you have to have this be your nucleophile. And then any active group here that could be coupling, you have to have that protected. So if you look at solid phase synthesis, the way that this is done, that you would start with this, you put this on this bead, you buy this, it's already protected here, it's already activated here. You don't find this in nature. And so this is protected. And so now what you do is you have to deblock, you have to take off the DMT group. So you take off the DMT group. And then what you do is you take this amidophosphate and this phosphoramidite. And so this alcohol now can attack that phosphorus because it's loaded while this one is protected. So now it's attacked that. And so now what you got to do is now you have to couple all the ones, you have to cap all the ones that didn't couple. You have to go through the capping step. And then what you do is now you take off this group, and now you have your next group, and then you go through the cycle again. You have to go through all these steps just to hook on one. This is what happens in a DNA synthesizer. Nobody knows how you do this without being able to block and deblock. Enzymes can do this, but no one has it. Now, you think of RNA. RNA is unstable because this is right off the Sigma Aldrich website. Because this hydroxyl will attack that phosphorus and off it comes. It's self-decomposition. That's why when you buy it, it's stored at minus 80 degrees, and it's stored in a buffer solution, and it's stored with chelators that grab any divalent salts. If you have salts in here like magnesium salts, this is even faster. This is why even people like Jack Szostak, these experts in RNA, who are origin of life researchers, say that if you're dealing with RNA, you only have minutes, not even days. You only have minutes when it's at room temperature of stability, only minutes. Yes, you can stabilize this enough to give mRNA injections, but remember, that's kept cold, and it's kept in a buffered solution, and it's kept stabilized. If remember when the Pfizer vaccine first came out, they said you had to store it at minus 80. Well, what's happening is they can turn it over so fast, you can store it at 0 degrees instead of minus 80, or either 0 or minus 20 in a freezer, minus 20. So you can store it at minus 20, but that's in a buffer, and that's with chelators. With great trouble, you have to stabilize these things because they would fall apart. RNA is really unstable. So when people talk about the RNA world hypothesis on how these things came about, that's a bunch of utter nonsense. When you think about the stability, you only have hours to work with these things. Lipids and the cell membrane. You think lipids are simple? They're not. So if you look at a lipid, you have a homochiral or stereogenic center. So this will start with glycerol. You have to, the two groups on glycerol are enantiotopic. You have to choose one of these hydrogens over the other, one of these side chains over the other in order to attack this. Once you put a group here, this now becomes a stereogenic center in that as soon as you put one group on one side, this is now going to become a stereogenic center once these two sides are different. So this is a big problem. You have a stereogenic center in every one of these phospholipids. You can't deal with a racemic mixture. Doesn't work. So people think that, oh, well, these evolved. You know, you can't just throw everything at the doorstep of evolution and say it took care of it. No, this had to happen before there was any biology. You had to have this because you have to have a well-ordered lipid bilayer in order to have a membrane to protect your cell. Whether it's a eukaryote or a prokaryote, you want to talk about a bacterium, fine. Bacterium has quite a sophisticated lipid bilayer system. It has either a glycolipid, thick layer, a gram-negative, and then a bilayer, or it has a bilayer and then a glycolipid and then another bilayer. Sphingolipids have 4 possible stereoisomers, not just 2. And then they form this membrane where you have the polar on both sides and this non-polar in the middle. So the summary, when you're addressed to origin of life researchers, they have no control of the absolute stereochemistry, the homochirality, little to no control of relative stereochemistry, no accounting of yield, no accounting of purification, no accounting of mass transport through the sequence. There's enormous human intervention at each phase. A prebiotic Earth would have none of these luxuries. Reading the articles on origin of life, it leaves enormous holes which render the work prebiotically irrelevant and misleading to the non-synthetic chemist. This is not for lipids only, it's for almost all origin of life researchers' syntheses. All right, chiral-induced spin selectivity. Some people say homochirality came later. You didn't have to have it with only one enantiomer. That is wrong. We're learning that because of something called homo-induced spin selectivity. This is not a bunch of esoteric stuff. This is real, and you have to deal with these problems. So how does nature do things so clearly? It's because they go through chiral environments. So you can take hydroxyl radical. Hydroxyl radical can go through a triplet surface and form oxygen and protons, or it can go through an anti-parallel singlet surface and form hydrogen peroxide. How does nature distinguish between these two paths? It has it go down a chiral environment, which is a spin selector. So what happens is you can have electrons have spin up or spin down, 2 different states of electrons. When we do chemistry, we normally just do whatever, we use both spin states, not nature. We've just learned this in the last 20 years. This is what we've learned in chiral-induced spin selectivity. And I've been promoting this and trying to show this to biologists. This is a huge, huge problem for origin of life, because what happens is in natural systems, they're what are called spin valves. They selectively choose out one of those 2 electrons and not the other. And so when you match the chirality of the system that it's going to run down with the spin of the electron and it's matched, there's very little scattering. And so there's very little heat release. If you have the wrong electron, you get scattering and you get heat. This is why biological systems don't burn up. This is why we can think, and we're only putting out about 3 watts from our brain, and we're not burning up with all these calculations going forth in our mind. It's because of chiral-induced spin selectivity. You can't get around this. You can't just say that this is esoteric. This is real stuff. And so what happens is that electrons with the right spin get through electrons with the wrong spin going down a chiral environment. So it will select for this. And so this happens. This is how plants survive. Photosynthesis. The most common synthetic reaction is photosynthesis. A magnesium atom sitting in the middle of a porphyrin, a photon comes in. That ejects an electron. How does that electron go hundreds of angstroms of distance without scattering? It's because it matches an electron spin with a chiral protein that it's going through, and it matches that. And this is how these things work. Without that, you'd have collisions and you have a lot of heat. This is what we know from chiral-induced spin selectivity. So might homochirality have come after cellular life started? There is no evidence for claimed racemic mixtures, meaning no homochirality being usable in cells. They've never been shown to function based on the reaction specificities, yields, and heat management that would be needed for cellular systems. Vast numbers of electron transfers take place in lipid bilayer membranes alone, so it's baseless to summarily dismiss the need for lipid homochirality. How did the proteins noted above become homochiral when there is no prebiotically relevant route ever shown to make amino acids in homochiral form or to separate the enantiomers or to polymerize them? But like so much that's promoted by those speaking about chemistry's life origin, there are incorrect claims that make the chemistry of life appear easy. All right, the assembly problem. So now let's just say somehow they got all the, how do you take those components and make a cell? Well, a protocell is a self-organized, endogenously ordered spherical collection of lipids proposed as a stepping stone to the origin of life. So you can make these spherical systems. This is not a soap particle. Some people confuse this with soap particle. It is not. It is not. A protocell is not like soap. Soap just doesn't have a lipid bilayer. It just has one layer. It has hydrophobic ones in that grab the grease, hydrophilic out that wash away with the water. When you have a vesicle, that means you have a lipid bilayer. It's water on the outside, water on the inside. Polar head here, bilayer. So you can make these, but they have to be made under a shearing environment. All right, you make lamella, and then you put it under shear. You can make these. There are little machines that can make these. It is not like soap and water. And so here's what happens is if you look at a lipid bilayer of a cell, it has a different constituency on the outer versus the inner side. Nobody ever makes protocells like that. Why? Because we don't know how it's done. Once it's in a cellular environment, there's something called flip-based enzymes that can control this. But the outer and the inner side have to be different in order to control the proton gradient so that you can have a proton gradient across this face. People who make these so-called protocells just dismiss this. You have to have all these organelles spanning them, all these features spanning these systems. Like you have these not organelles, but you have these usually protein or ionophore based systems that allow certain things in and certain things out. To control this, you have to be able to allow certain things in, certain things out. And now if you go to eukaryotic cells, then you have bilayers, lipid bilayers, and sometimes dual lipid bilayers among the internal organelles. If you have a eukaryotic cell, prokaryotic cell, it is simpler in the sense that you don't have these internal organelles, but you still have to deal with a very sophisticated lipid bilayer. And so then also you have to deal with, so that's part of the problem of just making the lipid bilayer. How do origin of life researchers deal with this? They don't deal with all the problems that I just told you about. All right, the interactomes. This is again one of the newer things that we're learning. This is the non-covalent connectivity within a functioning cell. So you have covalent bonds hooking together, and you have non-covalent interactions like hydrogen bonding, like dipolar interactions between a carbonyl on one and the oxygen of a carbonyl on one and the carbon of a carbonyl on a neighboring molecule, or you have pi-pi stacking, or you have lipophilic interactions between the nonpolar domains. Collectively, those are called van der Waals interactions. All of those put together, they're called van der Waals interactions. Van der Waals interactions, and I know I'm taking many of you doctors and physicians that are studying and bringing you back to your organic chemistry days and how wonderful that is. So that what happens is that you have non-covalent interactions. These non-covalent interactions are very important. As you know, it's non-covalent interactions that often give these proteins their tertiary structure, their secondary and tertiary structure. The primary structure is their sequence, but the secondary and the tertiary structure, this is all by these non-covalent interactions. But then you have molecule interacting with molecule by non-covalent interactions. These non-covalent interactions turn out to be really important to the ordering of a cell. That's why if you dehydrate a cell, take out all the waters of hydration, you can never reconstitute that cell once you've taken out all the waters of hydration, because you lose that order of alignment. This is why when a cell divides, it takes this information, puts it on each side, and then it divides this way. It keeps that order. Now, if you look at this, nobody knows how a viable cell emerges from the massive combinatorial complexity of its molecular components. And of course, nobody's ever synthetically mimicked it. An interactome is the whole set of molecular interactions in a particular cell. If one considers all the protein-protein interactome combinations, this is the non-covalent van der Waals attractions between them, keep them ordered, in just a single yeast cell, which is a very simple cell, the result is an estimated 10 to the 79 billion combinations. This is a crazy big number. 10 to the 90 is the estimated number of particles in the universe. You say, by particles, do you mean atoms, or do you mean electrons, protons, and neutrons? I mean, what do you mean by particles? It doesn't matter. The difference between the number of atoms and the number of electrons, protons, and neutrons is only a factor of, say, 20 or 50. That's a meaningless number when you're talking about something that's 10 to the 79 billion. This is a crazy big number that is so big, it's just mind-boggling. This is just protein. This is not protein DNA. And it's through these that the electrostatic potentials travel at near the speed of light. This is the electrostatic potentials where communication is traveling through. Origin of information. Critical for life is the origin of information, DNA or RNA. The information is primary, the matter is secondary. But we can't even get the matter. And why do I say the matter is secondary? Let me put it this way. I have a thought. I write down that thought on a piece of paper. All right, information has gone from my brain now to a piece of paper. The information is now on that piece of paper. It's on a different piece of matter. Now I type from that piece of paper into my keyboard. It goes into DRAM, dynamic random access memory. Now that information is in another form of matter. Now that information, I hit save and it goes into it goes into RAM. It's going to a flash memory. Now what happens is I upload that through the Wi-Fi system. So it starts uploading through an RF wave. That information now that was in my brain, that was on the paper, that was in all these different forms, is now in an RF wave. It's the same information. The information is primary. The matter is secondary. Same information, matter is now in that RF wave. That hits a box on my wall. That's now put into an electrons traveling through a wire. That then goes through some connection that goes to a server farm somewhere. That may go to a satellite, or it may go through a wire, and that goes to a server farm, again into another flash memory in a server farm. That's the cloud, the server farm. The matter is primary. The matter is secondary. The information is primary. The matter in which it is stored is secondary. So DNA and RNA are saying, we don't even know how to make those, let alone how to get the information that's encoded within it. All right, try to build a cell even hypothetically. All right, so a dream team, assemble a dream team and say, okay, people say, oh, well, you know, they found this molecule in outer space, so we can do it. They found traces of it. And even if tons were dumped on, they come as mixtures. How do you separate them? How do you use? But okay, I'll give you that. Assemble a dream team, all the smart people you got, and to make a living cell. And it has to have, you know, chemicals and homochiral form and all the informational code. Now, just to say, well, people will tell me, hey, Jim, you know, people have made synthetic cells. Well, in 2010, Craig Venter's team copied an existing bacterial genome and transplanted it into another cell. So he copied it. So if I go in and I take the computer chip out of my car and I go into my fabrication lab here at the university and I copy that chip and stick it into my car, would you say I made that car? No, I just copied the existing chip. Now, in 2016, Venter's team did something similar. They removed all but 473 genes from a natural genome and transplanted it into another cell. So what they did is they took a gene and they knocked out everything except for 473 genes, and they transplanted that into another cell. Again, the whole cell was already there. They just took a gene from one cell and made it bad by knocking out all but 473 and putting it into another cell. They didn't make the cell. They didn't make the interactomes. They didn't make the carbohydrates, the lipids, the saccharides, the nucleotides. They didn't make any of that. In 2021, what they did is they went back to these 473 and they added back in 19 because this cell didn't work very well. And so they added 19 back in, and so now that cell could reproduce. They didn't make all the pieces. They took existing pieces and did this type of thing. All right, so a challenge, make the simplest living cell. Just do it. A Nobel Prize would probably be awarded, and really quickly, if anyone could make a working cell, not a complex cell, even just a simple minimal cell that shows the functions of life. Do that and you win a Nobel Prize. All these people that understand origin of life, why don't you do it? Okay, reminder, what are the characteristics of life? It's got to be responsive to the environment, growth and change, ability to reproduce, have metabolism, breathe, and maintain homeostasis, be made of cells, passing on traits to offspring. That's what you have to have life. So these bioengineers have figured out you can do this theoretically. A minimum cell requires three main features: some form of metabolism to provide molecular building blocks and energy necessary for synthesizing cellular components, genetic replication from a template or an equivalent, and a boundary membrane. So this junk of just making a protocell, we made this thing and we packed it up with that does nothing. That does absolutely nothing. You have to have all of these pieces in there that are functioning. The necessity of the coordination between the, all right, let me get to this. So the smallest natural genome capable of autonomous growth or laboratory cultivation in pure culture and also in a defined medium is M. genitalium with 580 kilobases. That's 580,000 bases, or 580,000 nucleotides are needed. That's the minimum that's known. If you theoretically calculate, you say, what's the minimum that could be? Many people were saying 256. Later experiments said 206. You'd need at least 206 genes. All right, 206 genes. What is a gene? A gene is a distinct sequence of nucleotides. That's not 206 nucleotides. That's whole segments of nucleotides that you need at least that for a minimal cell. What's the minimal gene set going to have to include? It's going to have to be able to do DNA replication, repair, restriction, and modification, a basic transcription machinery, amino-acyl tRNA synthesis, tRNA maturation and modification, ribosomal proteins, ribosomal functioning, maturation and modification, translation factors, RNA degradation, protein processing, folding, and secretion, cellular division, transport, and energetic and intermediate metabolism, glycolysis, protein motive force generation, pentose phosphate pathway, lipid metabolism, biosynthesis of nucleotides and cofactors. The authors did not include rRNA or tRNA genes because they recognized that the basic substrate transport machinery could not be clearly defined, even though the minimal cell would rely on exogenous transport of the 20 amino acids coming in. So when people say, well, the cells were much simpler in the beginning, okay, they still had to have all of this. It still has to have all of this, or else it's not a cell. There's no life there. So we already know how simple it could be. This is how simple it could be. This is the calculated how simple it could be. All right, so now you got to make the simplest cell. First, you got to make all the monomers. You got to make the lipids, you got to make the nucleotides, you got to make the amino acids, and you got to make the sugars all in homochiral form. All the sugars, all of them. Go ahead, do it. Do it. You can't, right? You can't control the stereochemistry. You can't do this in any prebiotically relevant synthesis. Can't do it. And none of the syntheses have controlled stereochemistry. Okay, so what I'll do, nice guy, I will give you this. I'll give you all of these. Now just go ahead and make the cell. Okay, well, what you got to do is you got to polymerize these. Oh, okay. Well, how do I polymerize these? Well, I have to activate this and not act and have this attack this. But remember, when you have RNA, this is also an active site. How does this hydroxyl attack and not this one? Oh, it's a problem, because you can have the 2,5 linkages rather than the 3,5, and that acts as siRNA, interfering RNA. If you're making DNA, then you don't have to worry about that, but you still have to get the activation. So you still got to somehow figure out how to polymerize these. People have made short little things. I mean, like 6mers. I mean, it's hard to do this in a random, and that's without controlling any sequence. And remember, you're going to have the informational code here. How about peptides? How do you hook peptides together? Really hard. You got to do side chain protection in order to do the peptides. You have to have selective deep protection on the amine and the carboxylic acid in order to make peptides. It's hard to do. Carbohydrates, a gazillion different possible ways of putting this together. How do you take care of that? Okay, I'm a nice guy. I'll give you all those. I'll give you all those in polymerized forms. Okay, you got them. And I'll even give you with the sequence that you want. We'll take them out of living cells. I'll decompose the living cells. I'll give you all of these in the form you want. I'll give you the DNA. It's all there in polymerized form. It has the sequence you want. I'll give you all of this. Now just make the cell. You got all the components. Make the cell. Can't do it? Come on, a Nobel Prize awaits you. What's wrong? You're better than a mindless early Earth. What's wrong? Your dream team, all the groups in the world. There is not a chemist in the world, not a biochemist in the world that would say that they could take the components out of a cell and reconstruct a new cell. How do you make the interactomes? Nobody knows. So how do I, and we're also nowhere close to being able to do this. It's not like, well, we're just around the corner. No, we're nowhere close, because we can track our progress relative to the target of making a living cell. And what happens is the goalposts keep moving away. So in 1952, with the Miller-Urey experiment, some amino acids were made, all in racemic form, but people thought we were on the verge of making synthetic life. But what happens is every year we learn more about the complexity of the cell. So the complexity of the cell, we learn more, and it goes, it gets harder because of chiral induced spin selectivity. Wow, everything has to be chiral to begin with. Wow, that just got much harder. Goalposts moved way out. Oh, interactomes. I didn't know about interactomes. You mean I got to somehow get these to align in the right order when there's 10 to the 79 billion combinations? Oh, that makes it really hard. So the goalposts move way out. So we might learn a little bit, but the target of a cell is not just a bunch of protoplasm anymore. It's gotten so close, far away. So we're nowhere near solving this. So the next time you see an article like this, researchers solve the puzzle of the origin of life, don't believe it. It's just a bunch of nonsense. When you look up what they did, they took some scattered building blocks and they had it associate with a synthetic lipid bilayer. And then they say, oh, somehow RNA and proteins were made in the first cells. Huh? What they've done is they have bought a bunch of car parts, not made them. They took them from existing cells and they shoved it all into a car body. And they said, well, yeah, it's practically there. I mean, if you don't put each one of these piston rings around each one of these pistons and you can't just shove it in this car and say it's all there. And there's a lot more parts in a cell than there is here. And that's solving the puzzle of the origin of life when they got all their parts, not by making them, but by taking them out of existing cells, buying them, which the companies took them out of existing cells and put it into a lipid bilayer and said, there's your origin of life. That's what they did. So when they do this kind of thing, the public is misled. And so has the public been misled on origin of life? Yes. Here's a survey that was done by a friend of mine, John Narcombe. So they asked this question, and 80% of these people who answered questions had some form of a college degree, from an associate's degree to a PhD. Under simulations of Earth's early atmosphere, scientists have mixed molecules together in laboratories to produce complex life forms such as frogs. 37% of the general public thinks that that's true. Another question. Under simulations of Earth's early atmosphere, scientists have mixed molecules together in laboratories to produce simple, single-celled life forms such as bacteria. 68% of the general public feels that that is true. Not even close. Not even close to making a simple bacterium. Not even close. So is the general public misled? Absolutely. Have so-called scientific facts ever before been shown to be wrong? Yeah. Does the universe have a beginning? Scientific fact changed in 1964 with the steady state theory that was replaced by the Big Bang theory. In the 1950s, all scientists, they felt that the universe had no beginning. It always has been. But in 1964, it was nailed home that the universe had a beginning. So that changed in 1964. Scientific quote unquote fact that is not really a fact, but a theory changed. Darwinian theory to punctuated equilibrium. Scientific fact changed in 1972 when Eldridge and Gould proposed that the degree of gradualism commonly attributed to Charles Darwin, is virtually non-existent in the fossil record, meaning that in the fossil record, there were periods of rapid change and that were very short, and then nothing happens. Nothing happens for millions and millions of years, and then periods of rapid change. So it's not this gradual. So punctuated equilibrium changed this quote unquote fact in 1972. Climate change killed off the dinosaurs. That fact changed in 1980 when it became due to an asteroid impact, the Alvarez hypothesis, where an asteroid hit the Yucatan Peninsula that was iridium rich and threw up all this dust into the air that obscured the sunlight, and the plants died. So the herbivores died, and then the carnivores died as a result that followed off. And so the dinosaurs died off during that. So that fact changed in 1980. Random mutation and natural selection is suggested by Darwin and is taught as fact, are recognized by many evolutionists since the 1990s to be insufficient to account for the complexity of life. Now its neutral drift is quantitatively more important in understanding genetic differences between organisms. That's the changes that occur between me and my children and their children and their children. This just neutral drift that changes over generations. How long ago did the dinosaurs become extinct, or how stable is soft tissue? Well, scientific fact is being questioned since 2007 when Mary Higby Schweitzer, a paleontologist at NC State, led the group that discovered the remains of blood cells in dinosaur fossils, and later discovered soft tissue remains in Tyrannosaurus rex specimens. 2015, researchers reported finding structures similar to blood cells. These are not fossilized blood cells, but I'm talking blood cells and collagen fibers preserved in bone fossils of 6 Cretaceous dinosaur specimens, which are approximately 75 million years old. And lots of these now being found. People greatly questioned her at first, but she's been a hero in this. All right, now I'm not proposing God of the gaps, suggesting that which we do not know, boom, must be God. No, as a scientist, I would never say that we will never understand life's origin. I could never say that. One day in the distant future, we might understand life's origin and evolution of complex systems. One day we might understand that. But as of today, we don't. How does a complex system evolve? One system into another? We don't know. Evolutionists will tell you, well, one small change at a time. Well, whoop, that does me a lot of good. Tell me about that. Tell me about the details of how that happens. How does that happen? You know, you get into the details and everything falls apart. Origin of life, we have no idea. And we know that that's very far away because the target moves further away from us every year. That will not, if one day we should find an answer to this, it will not lessen God, but we will see him as all the more magnanimous. So if one day we find the answer to life's origin, we just say, wow, God, that's how you did it. You're amazing. It's not going to lessen God. Just like understanding that you know, you ask a person in the 1700s, how come two parents that are tall have tall offspring? Well, now we know something about that because of the discovery of DNA and how this information is stored and this information is passed on. That doesn't lessen God that we understand something about DNA. We look at God and we say, oh, so that's how you did it. That's amazing. What wonderful chemistry. So scientific fact versus the Bible. A scientific fact, water, H2O, has 2 hydrogen atoms and 1 oxygen atom. That will be the same throughout the universe, and that will never change. That is a fact. Now, we know it can have deuterium, it can have tritium, and so that you have, I think it's like 18, and you can have oxygen 16, oxygen 17, oxygen 18. So you have a number of different possible isotopomers, but it's still H2O, two hydrogens and an oxygen. Maybe different isotopes, but two hydrogens and oxygens. That's a fact. It doesn't change. There has never been discordance between scientific facts and a statement in the Bible, so there's no need to reconcile them. There are scientific theories, but not scientific facts that are in discord with the Bible. So-called scientific facts, which are really theories, are constantly changing, even on the order of decades, and certainly on the order of a century. So trying to twist the Bible to fit with a scientific theory is a frustrating endeavor. It's hard to do that because things keep changing in science. So do not let professors with their bold claims of facts upset you. Theories or conjectures are not facts, but unfortunately and shamefully, many professors themselves do not make the necessary distinction. This leads to the confusion of generations of students, and even professors themselves are confused on this, because I see it all the time. To the student inundated with misinformation, here's what I'm going to leave with you. Deuteronomy 13, verse 3 and 4 says, "You must not listen to the words of that prophet or dreamer. The Lord your God is testing you to find out whether you love him with all your heart and with all your soul. It is the Lord your God you must follow, and him you must revere. Keep his commandments and obey him. Serve him and hold fast to him." He's testing you. Are you going to hold fast to him? Are they presenting you with a scientific fact when they say that something is wrong in the Bible, or is it a theory? Theories change all the time. We are clueless on the origin of life, absolutely clueless. And I will go toe to toe with anyone on that, with anyone. We're going to leave you with a couple of verses. For those of you who do not believe in the physical resurrection of Jesus Christ, the Bible says, "I, even I, am the one who wipes out your transgressions for my own sake, and I will not remember your sins." This is God speaking. I, even I, am the one who will wipe out your transgressions for my own sake, and I will not remember your sins. God has come to forgive you. He has come to love you. What does it mean you're going to wipe out my transgressions for your own sake? I liken it to this. I have 4 children. If one of them were to go to jail, I would immediately go and bail them out. And if they've said to me, "Dad, I deserve to be here for what I've done," I'd say, "If it's in my power, I'm getting you out of here. I will mortgage my house if I have to get you out of here. For my own sake, I'm getting you out." And God says, no matter how evil you think you've been, for his own sake, he loves you enough to get you out of this. That if you confess with your mouth that Jesus is Lord and believe in your heart that God raised him from the dead, you will be saved. Give me an opportunity. If you do not believe in the physical resurrection of Jesus Christ, give me 1 hour of your time. You send me an email right here. It is an invitation to hear how I became a person of faith in Jesus and believe in his resurrection. You send me an email, tour@drjamestour.org. I will set up a Zoom conversation with you, and I will share with you. Give me 1 hour of your time. If you do not believe in the physical resurrection of Jesus Christ, and you want to hear why I believe in that, you want to hear that, let me tell you, and you will go away after that 1 hour, also believing in the physical resurrection of Jesus Christ. I see it all the time. You want to connect with me? Tour@drjamestour.org. You want to see some of my YouTube videos? It's DR James Tour. So you just go to YouTube, type in DR James Tour, the YouTube channel will come up, and you can connect with me. And with that, I wrap it up. I'm going to stop sharing my slides, and we can open it up for questions.
Thank you, Dr. Tor. Thanks so much for serving us well. Not heard many of those critiques against evolution. Personally, I'm a little rusty on my solid phase peptide synthesis, but I was keeping up when you said the public has been misled, and I wanted to ask a question of clarity before we get to our.
Can I make one point on what you said? I didn't address evolution
Yes.
at all. I was addressing origin of life, which is pre-evolution.
Thank you. Helpful distinction.
Okay. All right.
Yeah. So if I understood you correctly, it sounded like you were saying only a few people really in the world, a handful of people in the world, have a well-informed idea of how life could have begun on a molecular level, and those folks have overstated what has been done in laboratory experiments. That information somehow has worked its way into textbooks, but the people teaching that material, the people teaching the textbooks, they often don't understand what they're talking about.
No, I don't think you understood. No, I don't think you understood me correctly.
Yeah,
nobody in the world understands how life formed. Nobody in the world even has a good idea on this. Nobody. People project as if they have an idea, and they put out these false views of some prebiotic soup, lightning strikes, molecules come together, and molecules evolve and go toward life. People project as if they understand, but as soon as I start questioning them or they start getting questioned on the details, they get very readily lost. They project as they do, they publish papers, and then these get translated into the press with the author's excitement behind it that the origin of life researchers solve these mysteries of the origin of life. Those enter textbooks, and poor high school teachers, poor university professors think that somebody understands and then project that out to students. But nobody understands.
Okay. It's fair. One question that came in, I think you've answered it. If you want to add anything, please do, because it is an important question. Grace asked, if you use the complexity or fragility of chemistry, the stuff nobody knows, as evidence of a creator, does our view of God not shrink as science grows? So if hypothetically abiogenesis was true, would that mean God didn't do it?
No. That's exactly what I was saying at the end, that even if we find out, and I could never say that we won't, I presume one day we'll find out how life originated scientifically, that this is how it came about. That does not lessen God. That does not make God less, in my estimation. Now that I understand about DNA and how it do, I'm just amazed that he has encoded this information in the strings of molecules using 4 letters. I mean, just amazing. 4 letters, he's encoded all of this information, along with all the start and stop information, along with all this healing information. You get a wrong base in there, it's a real problem, but you have all these enzymes that go in and repair it. I'm like, God, you are amazing. So there's all this error correction, and then there's this duplication. How do you, in information science, what do you do to maintain the fidelity of a message? Well, you duplicate it and duplicate it and duplicate it, so it's not all dependent on one electron. Each bit of information in the transistor is based upon thousands of electrons. And why do you do this? You need duplication. God's already figured this out. So it makes God all the greater in my mind. The more I learn about science, the more magnanimous God is in my mind. I mean, this is just so I can see so much more than the man or the woman on the street can see, because I understand any material I look at, I know what it's made out of. I'm like, wow, this is amazing. And I look at a tree and how it's so strong. And why can you run your car into a tree and the car is destroyed and the tree just stands there laughing? What is it about carbohydrate strands that are hydrogen bonded now so they can give a little bit, that they don't just crack like some piece of plastic that you put up there? Because God's figured this thing out, and it's amazing the way God's figured this thing out. So it makes God all the greater. So I hope that answered your question.
I think Grace is satisfied. Thank you.
Hello, Dr. Chor. This is Janie. Grant actually lost his internet. So okay. I'm going to propose a question here, just the tail end of a question from Lori. What do you think will convince the secular world to accept the possibility that God is a reasonable explanation for the complexity of life that we see?
Okay, that's a very good question. And I'm going to give you an answer that you probably are not used to hearing. So Jesus said in John chapter 7, verse 17, so Jesus says, if anyone is willing to do his will, he will know of the teaching, whether it is of God or whether I speak from myself. So if anyone is willing to do his will, he will know of the teaching, whether it is of God or whether I speak from myself. So as I get to know Jesus, as I get to know him, I trust the Bible more and more. I trust the descriptions of God. I trust God more and more. As we do his will, we know of the teaching. People come to the Lord generally because of something that hits them in the heart. I didn't come to the Lord through science. Most people come to the Lord, something is working on their heart. I saw the famous Nobel laureate, Rick Smalley, the discoverer of C60, who's a friend of mine, come to the Lord. It hit him in the heart, not in the head, not through science. It hit him in the heart. That's generally how it does. The Bible says you must confess Jesus as Lord and believe in your heart that he's risen from the dead, and you will be saved. That's it. You don't have to believe in Adam and Eve. You don't even have to believe in the virgin birth. It may bother some people, but it says this is what you got to do, that Jesus is Lord and that he's risen from the dead. Once that happens, then people get to know God and they get to know his word and they begin to trust him and they begin to understand. So people do not come to know God and immediately throw out lots of science that they felt was facts. Never happens. Never happens. You get to know Jesus is Lord and he's risen from the dead. And the amazing thing about the resurrection, that's a very easy thing to communicate to people and they believe it. Every week, I see somebody come to know the Lord and believe in the resurrection of Jesus Christ, go from not believing to believing in the resurrection in a 30-minute conversation every week. Most of these people have PhDs because that's the world in which I work. And you say, how can that happen? Because the truth of the resurrection has been painted upon their hearts already by God. Because he has made that as a requirement for their salvation. He must have done something if he's going to make that as a requirement to make it believable, even to the PhD. Once that happens, then as they're willing to do his will, then they will know of the teaching, whether it is of God or whether I speak from myself, Jesus said. Then they begin to, their mind begins to become open and they say, wow, do I really understand how this whole thing works? No, I don't. And this is what I see, that when people come to know the Lord, like Rick Smalley himself, he came to know the Lord and he started believing just like, he says, we have no idea on the origin of life. We're clueless on this. Well, when he wasn't a believer, he had the whole idea on how this worked. So the thing is, we introduce them to Jesus Christ and his resurrection. And once that happens, then there's this gradual process of knowing. Okay.
Thank you. Here's another question. How would you advise Christian physicians and scientists in training, students, to effectively engage their teachers and mentors who hold on to many of the establishment ideas they point to as evidence for non-existence of God?
Non-existence of God. Okay. So, you know, I ask questions and it makes people feel really uncomfortable. I don't tell them this. I just ask questions. Tell me how that works. Tell me how one complex organism changes into another. We're not talking about small changes, but how does a respiratory system, how does some system, a complex system, change into another, to an optical system? How does that happen? What's the root? If life started from a single cell, because cell is a hard thing to do, how does that evolve such that you get the complexity and the diversity that you need? We didn't even just talk about, give me the details of that. What you will find is that it's storytelling, the best storytelling in the world. And then you give them a different fact, and they will then tell a story around that fact. And I probe that a little. Tell me about that story. You just said that such and such happens. Give me some chemical details around that. And you will see that these things start fading. And you can ask questions. I can ask a question at the board, a student is at the board, and I can have them melt in an instant. I can have professors get very frustrated with me very quickly, just by asking a few questions. I don't ever try to embarrass them. I just try to get them to think about what they're saying. So when a student is presenting something, they say, where do you think that comes? Well, that happened evolutionarily. So, okay, give me some details. What do you mean it happened evolutionarily? That term is bantered about. And we all have to say, oh, evolutionary. Ah, now I understand. Okay, proceed. No, I don't understand. Explain to me how that happens. And you will see how rapidly people's things begin to fall apart. So that's kind of what I see. But really, that's not what gets to the person. What gets to the person is the truth of the gospel. I know scientists and physicians, they don't like to hear this, but that's it. And that's why Paul said, it's Jesus's lordship and his resurrection. And that's what I try to get people to start thinking about. The resurrection of Jesus Christ, because our faith is not built on these mysteries in science. Our faith is built upon the resurrection of Jesus Christ. And the believing on the resurrection is easier than all these other things, because God has instilled that within the heart of the human being.
Okay. Thank you. Thank you very much. Just one real quick question. Have you a video discussing the truth of the resurrection?
Yes, I do. It's on Dr. James Tour on that YouTube channel, and it's I think it was the veracity of the biblical account of the resurrection of Jesus Christ, something like that. And it was a video that I did with a big audience back in the days when you could have a big auditorium with people all sitting next to each other. Remember those days? And and so I think it was filmed in around 2016, and it was filmed at Rice University by the Chi Alpha group. But that's on there. If you did resurrection, if you typed in that keyword Dr. James Tour resurrection, it would come up. There's one that I did, which was more of a Bible teaching on the resurrection. There was another I did more on the veracity of the claims of the biblical account of the resurrection. And the year before me, my friend Nobel, when he was alive, talked about the resurrection as well, from a different facet. And that you can find on the not on my site, but it's on the internet as well. So that'd be Nobel Qureshi speaking on the resurrection at Rice University. Those would be the key words.
Thank you very much. Here's Grant.
Thanks, Dr. Tor. My internet completely shut down. Sorry.
Yeah, and Grant, it's not doing very well now. Grant, it's not doing very well now. Your picture is frozen. If you shut off your video feed, then maybe we'll get your sound, because your sound is breaking up.
Let's try it this way.
Okay.
Okay. A couple questions that have come in. If you already asked this, let me know. But as far as reading material you'd suggest, as someone who wants to dig deeper and be a little more established. Apologetics was also mentioned.
Yeah, I read the primary literature in science, so that's usually not very helpful to most people. I have written, I think, 4 or 5 articles about origin of life for the journal Inference. So if you just Googled James Tour Inference, that is a free online journal, and you could read it there. And I cite the primary literature as I try to dissect a lot of the problems on origin of life. As far as an apologetics argument, there are good people out there like Stephen Meyer of the Discovery Institute and things like that. But personally, even though Stephen is a dear friend, I never quote him or Doug Axe on these sorts of things. I just go right back to the primary literature. That's just the world in which I live. I have never considered myself an apologist. I didn't even know what that word meant until a few years ago. And I just call it like I see it and just talk about chemistry, and it just nails people. And so I just talk about my chemistry, but those articles that I've written would get you started on this.
And your website does have a plethora of resources. So for those who have asked, feel free to go to Dr. Tour's website. You can easily find it on Google.
Yeah, it's jmtour.com, jmtour.com, or just Google Jim Tour and it comes up. And there's a section called personal topics, and you could find something called evolution creation. And I've written some I've written some there on that topic. And so, you could find more there.
Great. Did I understand you correctly when you referenced the Miller-Urey experiments that basically in the last 70 years, our studies, our understanding of origin of life has not improved?
Yeah, that I've said that over and over again. In Origin of Life, people have contested with me on that. And they say, okay, so how much closer are we to making the cell? Go ahead and make the cell for me. I'll give you all the components, make the cell for me. And then everything stops. Everything stops. And they have the RNA world hypothesis and say, okay, take your RNA, put it in a vesicle and tell me how far that takes you. How long is that RNA going to last? Tell me, why don't you do the experiment? You got a wonderful RNA world hypothesis. Do it there. RNA, they'll say, well, RNA it's an enzyme. You get a ribozyme, it duplicates itself. No, it hasn't. I mean, the only duplicative work that's been done with RNA is very well designed RNA that's primed to do duplication, and only 10% of it at most got replicated. Only 10%. That's not replication. And that 10% was never big enough to get replicated. So it's never done replication. Replication means, and that's under the very best of conditions. So just these things just fall apart. But I did a whole series on Origin of Life on Dr. James Tour. It is a 13-part series that goes through Origin of Life, that takes each one of these topics I talked about, and it's a 10-hour teaching over 13 different videos that just dissects this thing, and you can go through it and you'll learn a lot. And if the nice thing is that most of the people on this line, physicians and medical students, you've taken enough chemistry that you will be able to understand what I'm talking about.
Sounds like a fun Bible study. I hope a lot of our people take you up on that. We are going to wrap up. Please check out Dr. Tour's website. I've been there many times and looked at several of the resources, and it's one of the reasons why we're on this call right now. We also, of course, CMDA have a website. Love for you to check us out there. Join with us if you're not already a member. For more information on science and faith, the Discovery Institute Center for Science and Culture is a wonderful resource. And of course, partly this is brought to you by Erskine Seminary. So for advanced theological education, please check them out. Dr. Tour, would you like to say anything as far as closing us out? Any final encouragement or info?
Well, I don't know if it's allowed. Can I pray for the folks on the line?
It's allowed, please.
Abba, Father, I thank you so much for these people on the line, and especially the young people that are just starting out their careers. Father, protect them, watch over them, watch over their families. I pray, Lord, that they would find their security and strength in Jesus Christ and in him only. And through that, they would be able to have strong families and strong relationships. And Lord, I pray that they would not trash their families for their careers, but they would hold these things together and they would walk in your ways. Lord, your mercy upon these young people, I pray, your mercy for the glory of Jesus and in his name. Amen.
Amen. Well, God bless you, brother, you and your family, your work at Rice, and these kinds of talks. They are unique, and I hope you please keep doing them.
Thank you so much. Thank you for the invitation. If you like the content that's coming out on this channel, I've not monetized it in the sense of advertising, but if you want to give and you want to help support it, you can give to a 501c3, so it's fully tax deductible, and you can see the link below. We'd love to have your participation, and there's several mechanisms by which you could give. Thank you.